The next chapter is going to be chapter 16, which is how populations evolve. And this is going to be a continuation of just basically evolution and more specifics within how the mechanism of evolution actually works. So let's take a look at chapter 16.1, and we're going to get our really first definition of evolution. And this is when there is a change in the allele frequency of a population over time. There's a lot that goes on here. So change, we know what that is. Something's going to change. Allele frequency. Remember, we have like big B, little b. These are alleles. Frequency, this is how often something occurs. Population, same organism, same place, same time. Over time, so over generations. So this is how allele frequencies, or how the alleles are going to change in populations. Now, this is a famous picture that shows a gene pool. You can see all the alleles from all the organisms in a population. You can see all of the big B's and little b's in that population. We're talking about all of them. Now, with this section, we're going to be talking about something called Hardy-Weinberg, and there is some math here, but it's really not that bad. You can see our allele frequencies. So we can see the big A's and little a's in this first generation. It's about 50-50. You can see in this generation 10, there are more little a's just by visually looking at it. In this population, it's all little a's. So was there evolution? Did the allele frequencies change? Yes. So that means there is evolution that occurred. That's all evolution is. The allele frequencies changing from population to population, from generation to generation. This is why we can say evolution is fact, because we can watch these allele frequencies change from generation to generation. Now, in a population, we're going to talk about something called Hardy-Weinberg equilibrium. And when we talk about this, we have to understand that P is the frequency of the dominant allele and Q is the frequency of the recessive allele. Knowing that, we can kind of calculate the P and Q frequencies of both of all three generations here. So in class, I'll kind of take you through how we do that. But you can see, obviously, there's 50-50 here. It's about 75-25 here, and here it's 100% and 0% with the P and Q frequencies. We're also going to use these two. We're going to use these two equations when we talk about the Hardy-Weinberg equation. But basically, we're going to use these to calculate the frequencies of the big A's and the little a's and the dominant recessive alleles. When looking at these quadrants, you can see P squared is going to resemble, I'm going to go back here, the homozygous dominant genotype. The PQs are the heterozygous, and there's going to be two of them, so that's why we use 2PQ. And the Q squared is going to be the homozygous recessive here. So when we're looking at these variables, P squared, PQ, and Q squared, we're looking at the homozygous and heterozygous genotypes. Little note here, always start with the recessive genotypes. When we go through here, what we're going to do is we're going to be basically calculating for all 5 of these equations. I'm going to take you through more specifically in class, but there are a lot of good videos out there. I'm not going to take the time to kind of go over these because Bozeman Science does a great job of going over these equations. I'm not going to redo what he has already done. So what I would do is I would look through the Hardy-Weinberg Bozeman Science if you want a little bit more about the equations. But basically, when the Hardy-Weinberg equilibrium is met, this is when populations are not going to change. So you can see this equation. We have different percentages here. What happens is when the Hardy-Weinberg equilibrium is being met, all 5 of these conditions are being met. So for some reason, the alleles aren't changing. You can see there are no mutations. There's no migration. There's a large gene pool. There's random mating, and there's no selection. So when the alleles frequencies don't change, it means all 5 of these are occurring. No mutations, no new alleles are arising in this population. There's no migration into or out of the population. That means alleles aren't traveling. There's a large gene pool. Sometimes when you have small gene pools, things can happen. So it just gives us a bigger data set. Random mating, which means there's no determination of which phenotypes are going to be best suited for an environment. That should give you a little clue here about what's going to happen next. And there's no selection. There's no artificial or natural selection. So you might think like it's really hard to get all of these to occur so Hardy-Weinberg equilibrium is being met. Yeah, evolution is always occurring. That's basically what this equation is telling us. All right, that's where 16.1 ends. Our next section is going to be on something called genetic drift. And this is where kind of evolution, I don't want to say is random, but there's a lot more than just natural selection, artificial selection that can occur in a population. Our first example is going to be gene flow. This is the movement of alleles between populations. You can see these illustrations are just showing us, yes, alleles can move from one population to the next, and that's because immigration or emigration, and that's going to change the allele frequencies. Yes, this is going to lower the like the frequency of little a's here and raise it over here. So that is technically evolution. Genetic drift. This is more of a random how alleles change, or this is the randomness that can happen in population. It initially looks like this person specifically stepping on the green beetles, but this is just a random event. It's not like this person's targeting the green beetles here. So, did evolution occur? Did the alleles frequencies change? Yes. So this is evolution, but it's more of a random kind of way. If you think about, oh, can this happen? Take a look here. This is a boulder that randomly fell down. Um, did this change the allele frequency of the population? Yes. Was there a selection? No, it just randomly fell here. What if there was a really rare tree here that had a mutation? It's now not going to be there. So yeah, this is a random event that can occur that can change populations. Under this type of genetic drift, we have the bottleneck effect. This is where only a small portion of a population will make it to the next generation. And this is drastically going to change the population's diversity. You can see here the bottleneck effect is going to reduce the genetic diversity. And we see this because of natural disasters, diseases, loss of habitat, things like this. But this is really bad for a species because the genetic diversity is a good thing to have, obviously. But this is sometimes can just be random. Founder effect. This is when a small portion of a larger whole founds a new kind of community or area, and this can again rapidly change a population because the resulting population will be much different than the original population. What did the allele frequencies change? Yes. Did evolution occur? Yes. We're going to move into 16.3, which is the different types of natural selection. So let's take a look here. We have the stabilizing selection. You can see where our bell curve, we have this bell curve of variation of population. The outskirts here are removed or they die off more easily. The innermost portion here survives better. So this is called a stabilizing selection. And human birth weight actually follows this, where there's a higher death rate for babies that are born the very large, also with babies that are born very small. But in the middle, there is a high frequency of the baby surviving. So this is a stabilizing effect because the innermost portions they survive better, and or I should say more frequently, and the outermost portions don't survive as well. Now, obviously, with that, the medical community is definitely changing that bell curve. But before medicine, that's really what happened. All right, directional selection. This is where one side is going to be favored. You can see the rest is unfavored, so you can see the bell curve shifts. And horses are a great example of this, where the size has gotten larger and larger over throughout the years. So we can see how this body type has really just dramatically increased because of not only natural selection but artificial selection for humans. Our last type is called disruptive. This is when the inner is not favored and the outers are favored. This is going to create this kind of like two peaks, and mice are a great example of this because on dark colored rocks and light colored rocks, the mice will be favored. But in the middle, this middle type doesn't really blend in with either. So it's going to be selected against, which creates a population which has two distinct phenotypes it's called disruptive selection. Our last section in chapter 16 is going to focus on kind of other types of selection patterns we see in evolution. And the first type is called sexual selection. This is where adaptive changes are going to be selected for through males and females having choice within their mates. And the perfect example I like to give is the peacock's tail here. What we see is in their populations that the females will choose males with larger plumes and more of these. They're called eyes. So this is a selection pattern where uh, a larger tail is more beneficial for the peacocks to have. So over time, that's where these tails have really have gotten bigger and bigger. Darwin actually really struggled with this at first because he said that he couldn't really figure out why the peacocks have this large tail. This is because the peacocks have really no other beneficial, trait that comes along with these tails, and they actually can be caught with these tails, so it makes them almost detrimental. So why would they have these tails that would be make them easily caught by predators? It's because the females were selecting for traits of the peacocks that were a lot larger. Back in I always like to mention here, whenever Darwin first proposed this, he was actually kind of say mocked, but like people didn't accept that females had choice back in the day. They remember this is the 1800s. This is what they thought. They didn't really think that female choice was that powerful would be a selection process. Now we obviously know it is a huge selection process within certain species, and this creates this male competition. They did accept male competition, but they just didn't really accept female choice. And we now know there is a lot of species that also have female choice. And by the way, it also goes there in the opposite direction where it's male choice as well. But for example, the male seahorse actually will carry the will actually I don't want to say that would become pregnant, but they will carry the offspring. So there is also selection the other way, but it's almost like a dynamic between the two of males and females. And in species that we see this, we call them asexual. It's called sexual dimorphism, where there's going to be distinct features of the males and females within the species. You can see the lions, the peacocks, the ducks here, the monkeys here. I think these are baboons. They have distinct features within each of the phenotypes of the males and females. Like I said, with the seahorses as well. What else do we have? Other types of selection. This is called exaptation. This is where a natural selection evolves a previous like trait for a new use. So the example I give is the feather, and feathers were originally used not for flying, but for more of thermoregulation. When we increase the surface area, oh wow, we're going to use that term again. When we're increasing the surface area, we're going to increase the kind of how much the organism can keep heat in. So originally these were meant to be for thermoregulation and keeping warm, but as time went on, they actually found a new use. This is a feather now that we see today, but it's also used for flying. So it's not just that an organism will randomly create a new adaptation; it's that we reuse adaptations for new purposes. Um, one kind of other things that we're going to talk about is evolution isn't perfect. It doesn't create these perfect structures. An example of which is like the back of humans. Like the back is just misshapen. This is why we have so many back problems. Is we evolved to not walk upright. We walked on all fours millions of years ago. Now we're bipedal, which means we walk on two legs. We have so many problems because originally our backs were more parallel with the ground. So evolution isn't perfect. We're going to go over a couple of other examples in class of adaptations that they seem like they work, but they're actually kind of man. If we could change it, we would. But it's just how evolution has to work. Our last type of selection is called heterozygous advantage. This is when the heterozygous has an advantage over both of the homozygous genotypes. You can see big A, is susceptible to malaria, big A, little a. This is when the person has the sickle cell trait but not the anemia. This allows them to be resistant against malaria, which is a huge benefit to have if you're in locations like Africa where malaria is very prevalent. If you have sickle cell anemia, it's very deadly, but the heterozygous advantage here gives this trait a kind of a reason to be in the population. You can see the sickle cells here, what they look like. And again, when a person has sickle cell trait, they have half the cells that are sickle and half the cells that are normal cells. So they're heterozygous for it, not homozygous for it. You can see where malaria is, and you can see where the sickle cell trait is most prevalent at. All right, this is going to end chapter 16 for us.